CRISPR is a programmable precision gene editing system, a technology adapted directly from the natural immune defenses of bacteria. But utilizing this system to cure a hereditary disease presents a biological paradox. To fix a harmful mutation, we must first critically damage the DNA. This damage takes a highly specific form. It requires creating a precise, double-stranded break at an exact location on the genome. There is a common misconception that CRISPR cuts the genetic code. It does not. The mechanism only cuts the DNA. The entire potential of this intervention relies on what the cell chooses to do in the immediate aftermath of that structural damage. Clinical application relies on coupling this targeted molecular cleavage with the cell's endogenous repair mechanisms. To understand this system's precision, we examine two molecular components, Cas9 and guide RNA. Cas9 acts as molecular scissors, cutting the DNA helix. The guide RNA is a synthesized molecule programmed to match a specific genetic sequence. Before intervention, scientists must first isolate and identify the exact biological sequence of the harmful genetic mutation. Once identified, a custom gRNA is synthesized to perfectly complement that targeted sequence. Inside the cell, the gRNA searches for its match, binds to the complementary DNA, and acts as an anchor, pulling the Cas9 protein into position. Pairing a universal cutter with a highly customizable guide allows researchers to rapidly target virtually any sequence in the human genome. Firmly anchored in place, the Cas9 protein complex executes a targeted double-stranded break directly across the DNA helix. The moment that cut is complete, the active role of the CRISPR system is finished. As mapped out in this flowchart, the severed DNA forces a biological divergence. The cell's default emergency response is the pathway branching left, non-homologous end joining, or NHEJ. NHEJ rapidly forces the broken DNA ends back together without referencing a template. Because it operates blindly, this process frequently introduces unintended insertions or deletions into the genetic code. While technically an error, researchers leverage this sloppy repair therapeutically to permanently knock out a faulty disease-causing gene. Achieving true precision requires the alternative repair mechanism on the right, homology-directed repair, or HDR. To trigger HDR, scientists introduce a custom DNA template. The cell uses this supply template to flawlessly rebuild the broken sequence, replacing the mutation with healthy code. Achieving specific genetic correction requires prioritizing the complex HDR pathway, as the default NHEJ knockout is limited to gene disruption rather than sequence replacement. Sickle cell disease is a prime candidate for this type of intervention because it is monogenic. It is caused by a single, isolated point mutation. That specific biochemical target is a mutation residing in the HBB gene. This mutation alters the production of hemoglobin, causing the molecules to stick together and create rigid, malformed red blood cells that block blood flow. The clinical intervention begins by extracting blood-forming stem cells directly from the patient. In a laboratory setting, the custom Cas9 gRNA complex is introduced to these isolated cells, targeting the mutated HBB gene. Crucially, an HDR template is also introduced, forcing the cells to rewrite the sequence so they can produce functional or fetal hemoglobin. Infused back into the patient, these edited stem cells rapidly multiply, repopulating the entire bloodstream with healthy, flexible red blood cells, eliminating disease symptoms. Exploiting a highly localized biochemical repair mechanism allows us to functionally reverse a complex whole organism disease. Despite these molecular successes, overarching biological boundaries dictate how and when CRISPR can be ethically deployed. This minimalistic cellular lineage map illustrates the critical boundary of somatic gene editing. These treatments target mature body tissues of a single patient. They hit a definitive biological barrier. Somatic changes absolutely cannot be inherited by future offspring. This contrasts directly with germline gene editing, which targets embryos, sperm, or eggs. Germline edits integrate into the foundational blueprint, propagating downward into all future generations. Beyond the germline debate, approved somatic therapies face severe technical bottlenecks, notably the immense challenge of safely delivering CRISPR components into specific target tissues. There is also the persistent danger of off-target effects, where Cas9 may occasionally bind to and cleave unintended similar genomic locations. Furthermore, relying on the precise HDR pathway for sequence correction is statistically much less efficient than the cell defaulting to the error-prone NHEJ pathway. The distinction between non-heritable treatments and heritable changes, coupled with these efficiency bottlenecks, defines the regulatory and biological boundaries of modern genetic engineering. The workflow for precision genetic engineering relies on two distinct stages: gRNA for programmable targeting and Cas9 for structural cleavage. As seen in this sequence, the final genetic modification is dependent on the cell's endogenous HDR and NHEJ mechanisms to repair the break. The mathematical precision of the CRISPR design must always contend with the chaotic reality of unintended biological variables inside a living cell. Currently, the active frontiers of genomic research are focused on maximizing HDR efficiency and engineering new Cas9 variants to eliminate off-target cuts. CRISPR is not a flawless cure-all. It is an iterative, highly potent biochemical tool that requires rigorous testing and continuous monitoring. The ultimate goal of this technology is to neutralize debilitating inherited genetic conditions at their root molecular cause. Mastering these foundational biochemical principles is the requisite first step to actively rewriting the hereditary constraints of human biology.